Yeah, thank you so much for the introduction, and of course, thanks to the organizers for giving me the opportunity to present this story. And I hope that by now you're a bit hypnotized by the video. And so I'm going to tell you the story about this new experimental system that we formed in the Sapienza, and this is my latest project. And that is formed by a magnetic crystal, magnetic colloidal crystal, that is activated by light-driven bacteria that hopefully you'll see here that I painted the Sapienza color. They're actually not this red, but that activate this crystal. So in this talk, I'm gonna, like in the first half of the talk, I'm gonna explain a bit how the fluctuations, how active fluctuations differ from passive fluctuations or thermal fluctuations. And then in the second part, I'm going to talk to you a bit about how this crystal can melt. So, of course, I don't need to tell all of you why active solids and active crystals are interesting. But I did want to mention a bit of the motivation of this work. That is, some Brownian magnetic colloidal crystals. There were many studies in the late 90s and early 2000s, and all of these stories of how colloids can act as model systems, right? To study different transitions at different length scales. They've been used to study glass transition, melting transition, in this case, specifically the 2D melting transition, and also the harmonic behavior of colloidal crystals that compared to more atomic crystals. So the cool thing about these magnetic colloidal crystals is that they're very easily controlled with an external field, and that the interactions, of course, are relatively well known and they're easy to access. And of course, as any colloids, you can observe them in the microscope very easily. So that is why they're interesting to study. So our idea is, what if how can we make this crystal active? How can we find an analog of an active crystal that we can study some physical properties of active systems in this system. So this was our idea, and so to do that, I have to present to you one of also the main characters of this story. That is photokinetic bacteria. In this case, they're specifically Escherichia coli. They're, as I'm sure of many of you know, 2-micron-sized bodies, self-propelled. In the wild type, they do run and tumble motion. What's special of, in general, bacteria, they have this flagellar motor that is very powerful, so they can propel at the microscale, of course. But Escherichia coli specifically has a genetics that is very well known and very well studied, and thanks to genetical engineering, we can modify it almost at will, almost as if we synthesize a particle, and we can make it do almost whatever we want. So in this case, to have it photokinetic, it's genetically engineered to produce this protein that is called protorhodopsin. This is a transmembrane protein that is usually found in systems in the sea, in marine bacteria. But when E. coli are modified to produce this protein, this acts, if you want, naively as a nanosolar cell that captures the light intensity of the environment and then transfers it to the energy to the motor, and the more light that it captures, the faster the motor spins and the faster the cell moves. So that is why it's photokinetic. The brighter the light intensity that captures, the faster it moves. So they swim with a speed determined by the intensity of light from their environment, right? This is a couple of examples of, hopefully you can see that with dim green light in the top, they're swimming but not very fast, and in the bottom they're swimming much faster, right? So this is also a graph of a work that was done in the group that I was at several years ago, of the speed as a function of the intensity. Just briefly, a couple of cool things that can be done with this bacteria. You can paint with bacteria. They painted the Mona Lisa. This is going in a loop, so you can shape with digital light projectors. You can shape light intensity, so you can make it paint the Mona Lisa. On a, think a bit less interesting note, but also cool project that I did before this, you can guide, by projecting small spots of light, you can guide the bacteria towards, you can do a feedback loop with a camera connected to the light projector, and you can guide the bacteria towards the center and confine them there. The red part is the bacteria, the green part is the light that you're projecting, so this is what you can do.
Can I ask a question?
Yeah, sure.
So the light, when it activates the bacterial redoxin, is actually helping the bacteria make more ATP? How does it actually work?
Yeah, exactly. So you actually have to drown the bacteria, so there has to be no aerobic production of ATP. So if you take out the oxygen from the sample or you seal it, then it activates this mechanism that is a type of taxis. So, yeah.
And they have an analogy to chemotaxis. They will go towards the light somehow, the light gradient?
So yeah, this can also be genetically engineered. If I'm not a biologist, I don't know exactly, but there's a gene that controls the taxis that is different from the one that. So
thank you.
Thank you. So back at why we care about these bacteria, back to the crystals. So we use them as a bacterial bath. Right? It was also proven several years ago that if you put a passive particle in a dense bath of active particles, in this case, in a dense bath of bacteria, the motion of the Brownian particle or the passive particle becomes active, right? So there's a characteristic time, there's a ballistic diffusion in the beginning, and then there's just hotter diffusion at long times. So with these two things, what we did is we obtained a photokinetic bath with activity controllable via external light. This is very interesting because it allows us to have an extra degree of control to the system besides the magnetic field. So the experimental system, just to describe it to you, we have these paramagnetic colloids of 4.5 microns diameter. Without any magnetic field, they have no net moment. But then when you apply a magnetic field, they have an induced moment in the direction of the field applied. In this case, we apply it perpendicular to the sample. And of course, depending on the geometry, it can induce attraction or repulsion. In this case, we want it to induce repulsion. So to avoid the particles going on 3D, we confine the sample in 2D between two glass plates and apply this field perpendicular to the sample. So the particles self-assemble into this crystal, and then, of course, we add the photokinetic bacterial bath. As I explained, these Escherichia coli, they do run and tumble, and they are genetically modified to respond to green light. And then, of course, we have this, as I said, this extra degree of control of the green light intensity. So we have these two knobs, right? The magnetic field that controls the confinement and the interaction of the crystal. And then we have the green light that controls the activity of the bath and just acts on the bacteria. So a bit on the dynamics that we expect of these particles. In general, particles in a bath of bacteria are display what is called a motion of active Olsen-Nuremberg particles that you can model with the following differential stochastic equation, that it has a term of a thermal noise, and then it has a term of the active noise that accounts for the bacterial bath that has an active noise that is exponentially time correlated with a characteristic time tau. That is how the activity acts on these particles. And of course, we have the long-range dipole-dipole interactions. So in this case, the motion of these active Olsen-Nuremberg particles causes the displacement to be short-time Gaussian distributed. So this is an experiment with no field, so you can see. And of course, this can be simulated to compare with our experiments. My colleagues and collaborators, Nicoletta and Claudio, did this so we can compare our experiments and verify our results. So again, sorry. Again, this is the motion of a single colloid to characterize it with no external magnetic field. So we can, to understand what the parameters that we're dealing with, because there's many factors here, right? Bacterial density, etc. But what we care about is how bacteria affect the colloids at different light intensities, right? So these are the superimposed colloidal trajectories at different light intensities that affect the bath, that affect the colloids, without interactions or anything. So we can calculate the mean square displacement and fit it to the equation of active Wilson-Nuremberg particles that we expect, and we can obtain the terms of the thermal diffusion, the active diffusion, and the persistence time. So we know from each experiment what parameters are we looking for here. And it's important to note that we can go to almost 10 times the thermal diffusion just with some bacteria, so that is interesting. Okay, so this is our experimental setup. It's relatively simple, just an inverted microscope with a green LED that controls the intensity, and then the magnetic coil that connected to a power supply that controls the magnetic field. This is a photo of it. So this is the sample of, hopefully you see the bacteria in white swimming around a bit, and then you see the particles are disordered. There's no magnetic field yet. So and then if we turn up the magnetic field, it quickly self-assembles into this, hopefully you see it, pretty ordered crystal, right? And as I said, the magnetic interaction in these colloids is it's well known. So it's besides the prefactor, it depends on the squares and the square of the strength of the field and on the interparticle distance to the third. With this, at the first approximation, we can define or think of a dimensional global temperature that includes both the magnetic contribution, confinement contribution, and the active diffusion. This does not take into account any difference of any difference that the persistence time can make. We assume that thermal and active fluctuations are acting the same. So we have the active diffusion, which is controlled by the intensity of light, and this scaling of the temperature that is controlled by the magnetic field. So finally, here's an example of the same system with the green light off and green light on. Hopefully you can see that the bacteria here are, even though this is very sped up, are just performing Brownian motion, not really swimming. Here they're swimming around, hitting the colloids, and I'm sure that you can see that the colloids are moving much hotter, at least, than in the top part. So that is also a very interesting part of this system, that we can have the passive equivalent of the same piece of the system just by turning off the light. So we know that is the same particle, same experimental data, etc. Of course, the important question here, and the interesting one that we wanted to ask, is if there are any qualitative differences between these active and the fluctuations caused by the bacteria and thermal fluctuations caused by Brownian motion. To do this, we looked at the crystal's harmonic behavior when excited by bacteria or by the thermal fluctuations. This was done before in the same passive system without bacteria, and was used to study the harmonic behavior, as I showed you before, of these colloids, but we wanted to see what happened again with these active fluctuations, right? So we know that the harmonic potential energy of the crystal can be written as this. This is a dynamical matrix in Fourier space, and this is the displacement field of the colloids respect to their equilibrium position. From energy equipartition theorem and this, we know that in equilibrium, of course, every mode of the crystal provides the same measurement of the temperature, right? So we can think of it as a mode being like a kind of a temperature, and every one of them provides the same T. However, it was studied and predicted by some just theoretical work that this does not happen. This is not the same in active systems. Specifically in active Wilson-Nuremberg, they proposed a generalization. So what we do is we introduce a term of that we call active temperature. That does depend on the mode, right? So it depends on K in this case, and also on the parameter. So in this case, different modes if this were true, it would mean that different modes in the system prove different temperatures, right? Different modes have different temperatures. They don't have, it's not like in equilibrium that you provide the same measurement of the temperature. So we use these relaxation modes as a type of thermometers, if you want, to measure these different effective temperatures. And indeed, we find that here are some experimental simulations for different persistence time, which is the important parameter here. For relevant enough, high enough persistence time, we do find a mode dependency. So this is the active temperature scaled by Brownian temperature. So we see that in short persistence time, the characteristic is the same. It doesn't depend on the mode. It's just hot. It's just a hotter Brownian crystal. However, for persistence time that are important enough, it does depend. So it does have this multiple temperature effect, right? A mode-specific temperature. So you can think of this as active and Brownian fluctuations acting differently on the different system degrees of freedom, which is, in the end, not that surprising, thinking that we're out of equilibrium, but nevertheless, very interesting. Okay, back to the second part of the talk. So, yeah, sure.
So using the same argument, you would also expect you don't want me to throw the mic. So sorry, so following the same argument of the like non-thermal driving of your modes, you'd also expect a correlation length in your system. Have you looked at that?
Yeah, we're looking into that, but it's complicated with the experimental system size. But yeah, it depends. Yeah.
So because it should also the same scaling with the square root of persistence time.
Yeah. I mean, yeah, I have to think about it. Yeah.
Happy to talk. Yeah.
Yeah. Thank you. So if there are no more questions about this first part for now, I'm going to go into the second part. So we wanted to explore, of course, after exploring this harmonic behavior, higher temperature phases. So the first question is if the crystal can melt just through activity, through these bacterial heats. And the answer was yes. So this is with the same magnetic field, with the light off and the light on. Hopefully you can see that the system is more disordered and neighbors are starting to diffuse away from each other. So here it's just, this is particle trajectories, these are not cells, but it's a Voronoi diagram of a very small region of the sample, but just so you can see that defects start to appear when you turn on the light. So in the beginning the light of the system was off, all of the particles had 6 nearest neighbors, and then some defects start to appear. So you can characterize these. For now, we just looked at the defect fraction and the von Dörder parameter evolution with time, and that we see that in a couple of minutes or less, the system quickly loses this disorder. So now that we've seen that indeed we can have bacteria powerful enough to destroy order in the system, we wanted to do an experimental phase diagram using these two knobs of magnetic field and light intensity or a bacteria that controls the active diffusivity to understand what is happening in the system. We needed, of course, a melting criterion. We used one that was used in this paper in the 2000s for same system, again passive, which is the Lindman parameter, but in 2D, that depends on time, is the dynamic Lindman parameter. Just measures the cross correlation between nearest neighbors. So in the end, what it means is that if it's bound at long times, that neighbors are always together and the system is a crystal. If this parameter diverges, it means that the neighbors are diffusing away from each other and the system melts.
So just a quick question.
Yeah.
Given that you have access to all the complete configuration, why not just measure the structure itself? Because I mean, Lindemann is kind of a crude empirical criterion, and even in a crystal, in principle, actually, because of vacancy motion, particles do diffuse, albeit slowly. But objectively, a crystal is a crystal if it's got quasi-Bragg peaks or whatever it is, and it's not if it's not. So why not just do that?
We did look at the structure, but in the end we just wanted to do a thing that was because I'll come back to Lindemann after that. But we did look at the G6, and so we needed very big systems that sometimes in experiments we just had hundreds, because these are a lot of data, so hundreds of particles. And this is a local parameter that can be done in smaller systems. So we just separed the system between crystal and melting using this Lindemann parameter, and in here we just colored it so you can see according to the orientational bond order parameter. So with this, you see that we have two melting routes, right? We have the passive melting route, which is just decreasing the magnetic field, of course. This is the classical route. And then we have the active melting route that involves just increasing active diffusivity. Of course, again, as before, the question is if there's any qualitative difference between active and passive melting routes. After a lot of analysis and thinking and simulations, we found again that the difference lied in the Lindemann parameter. So it is so first, if we plot again this parameter that I remind you is across fluctuations of nearest neighbors. For the passive route, we find, so this is an adimensional parameter. We find the melting threshold parameter for Lindemann that is very similar, that is almost the same as the one that was, again, in the same passive systems. However, if we do this through the active route, we surprisingly find that the melting threshold is much lower, both in experiments and simulations, and that the system starts to diffuse away from each other at parameters much lower.
Yeah, so I just had a question, maybe you've already going to address this, but in the absence of activity, this is the best example I know of two-step melting with a hexatic phase and Georg-Maritz group, etc.
Yeah.
So you'll tell us how that changes when you put on activity?
I won't tell you. I didn't put it it's in the paper because it doesn't. We didn't see it change. Except for, I mean, as far as we studied, we didn't see any difference from equilibrium. Besides a shift on the melting temperature for very persistent.
But there's not one melting temperature. There's two. There's where you lose the translational order and then you lose the bond order.
Yes.
And this Lindemann criterion, as Shriram was suggesting, is fairly imprecise. It's not to catch such subtleties.
It's not?
It's fairly imprecise. If you have vacancies, as we just heard, you'll still have particles wandering around. They won't be localized, in fact. And so, again, maybe you need very big systems, as the group at Constance had.
Yeah.
But and there are interesting questions like the divergence of the shear viscosity at the hexatic to crystal transition.
Yeah, clearly.
And how that changes with activity seems to me a fascinating question.
Yeah. But as I said, in the range of parameters that we explored, we did not see any difference in any of the two transitions in the structurally. So
how many particles can you look at a time?
Hundreds, almost 1,000. Yeah.
Okay. Thank you.
Thank you. So again, we did see a lowering of this critical threshold of the melting of the Lindemann parameter that was accompanied with an increase of the melting temperature in this system, which a fact that is seems a bit counterintuitive. So to understand what was happening here, we wanted to take a step back and just look at a very simple representation of the problem that is just one particle trapped in a sinusoidal potential. in this case, an active, again, active Austin-Nuremberg particle trapped in a sinusoidal potential. And we measured the mean square displacement, and in the end, we did see the same effect rapidly, that in active systems, when persistence appears, the threshold is much lower, right? So again, looking at the data and performing a bit of analysis on this, we saw that the melting temperature, the Linman criterion again decreases as a result of rare individual persistence hopping events. This means that the particles, the fluctuations of the particles when activity is increasing are damped. This is an effect that is known for this type of motion in confinement. However, some of the particles have enough persistence and enough strength to hop off their minimum and trigger melting. So that is how we interpret it, and again, the data seem to be pretty consistent. So just time. The conclusions, so a bit of take-home messages that we experimentally realized this novel system, this active magnetic crystal composed of these paramagnetic particles in a bath of light-driven bacteria. We studied the active fluctuations and how these relaxation modes are different from thermal fluctuations, and in specifically short wavelength modes are colder than long wavelength ones. And then we explored the active melting route, and what we did see is that there's a significantly lower critical threshold of the Linman parameter in the active case. So I wanted to thank everyone that worked in this project, of course, you for your attention, and I would happily take any questions.
Thank you. Okay, let's start the questions.
Hey, great talk. An excellent system. I promised one question, so I'll ask one question. So it's related to the questions that were already asked, actually. So what about the critical exponent, right? You guys probably saw the critical exponent. Is it the same as in the passive case? Is it different? Does it depend on activity? Because this is something that I would expect. That you know, the activity shifts the transition, but the critical exponent will depend on its value.
Yeah.
So, but you know, I mean, you look at the correlation functions and the decay of the order parameter, right?
Yeah.
So in both cases, I would like to know what happens.
Yeah, I mean, in the end, as I said, we just saw a change on the melting temperature. So systems that with the same temperature are liquid, decays the G6 of R decays exponentially. With the same temperature, they're crystalline, right? Okay. But we did not look, because experimentally it's very hard, and in simulations there's many papers that looked at this. There's a couple that I can point to you at offline that did see a change of this exponent in similar
because
systems.
We have, okay, yeah. Sure, we'll talk later.
Yeah.
Great talk.
Thank you.
Really cool stuff. What I have in mind is probably a headache, but I don't know, maybe it might make at least a little bit of sense. What I like about this system is that the activity-induced fluctuations, you can see what's causing them, as opposed to a lot of microscopic systems that are ultimately from molecular motors that are hard to visualize. And so presumably you should be able to map the change in entropy from the Brownian case to the active case down to the trajectories of the individual bacteria and their hydrodynamic interactions with the colloids. Is there anything in that? Could that be helpful?
I mean, here actually you're looking at an image that is again half artificial, but on the left it's like the variance of intensity of the bacterial bath on time. So you can see that this was just to compare if bacteria were moving or not. But yeah, I mean, that would be another interesting way to quantify. I mean, we just looked at the particle trajectories to simplify a bit the problem, but of course, and also this leads to also many interesting questions of how the bacteria swim around this landscape, right? If they accumulate a bit more on the edges or not, yeah. But thank you. I hope that answered a bit.
Yes, thank you, and cool stuff again.
Thank you.
We got that side.
Yes, it's really nice. I wanted to follow up on what I said earlier, maybe be more specific. So if you use an argument a bit like the one you said with Caprini, yeah, you can derive a correlation length in the displacement field in your system, and that becomes longer and longer as your persistence goes larger. And there's actually also a recent paper of Hugues for crystals, which show this. Yeah. So I would expect that to mess with your melting, because you have that mesoscopic correlation length when you start to melt, so that might be responsible for your Lindemann criterion stopping to work.
Yeah, I mean, that is an interesting point. We did not think it as much as the Lindemann criterion stopping to work, but just that the fluctuations are different, right? So it just has another threshold. But yeah, we were wondering, an important question was to link the wavelength or the mode dependence to the melting, right? So in our mind, we naively thought that in Lindemann are the shorter lengths that were, so that is what is affected by, so maybe that is why, but yeah, that is definitely a future thing to look at to understand the system more. Yeah.
Thanks. Yeah.
Okay. So
in the introduction of your talk, you talked about you were able to concentrate these bacteria.
Yeah.
So that kind of means you have some sort of directional control over these bacteria. What would happen to the melting if you were to, like, what do you think that would happen to the melting if you were to do a similar? Yeah,
that is very interesting. This is also the next step to control the bacteria more spatially with, this leads to many interesting questions. Yeah, this is, but yeah, I mean, what is your idea to direct the bacteria?
Just like one direction, just to say, I'm going to point all these bacteria to go that way, and then what happens to?
Yeah, I mean that you could maybe create a grain boundary that is, yeah, at least, yeah, I don't know. We haven't. We're on it.
Oh, thanks.
Thank you.
Hey. I don't know if it's interesting, but just how did you choose the size of the colloids and like the bacterial density? And do you think that would change stuff?
The size of the colloids, there's these commercial ones that I know they work because I worked with them before. Also, I wanted ones relatively larger than the bacteria, so it could not affect as much as one. But yeah, I mean, we did not have any other particles, but that would be interesting. And then the bacterial density, you can play a bit around it to change the parameters of active diffusion and tau, right? So, and also if you want to go much more to an extreme, you can, that we haven't explored that yet, you could even go to bacterial turbulence regime. Then the dynamics would be, I think, completely chaotic, but yeah, that would be interesting.
Very cool talk. So it's a clarification. I'm sorry if I missed this, but you showed that equipartition is violated, and you have these active temperatures that depend on the wavelength.
Yeah.
So these active temperatures decrease as the wave vector, the wave number increases. So, right?
It's in this reciprocal space, so it's the opposite. But yeah.
Wave number increases.
So yeah, the mode.
Yeah.
Yeah.
So meaning that shorter wavelengths have lower active temperatures, right?
So longer, sorry, I'm a bit dyslexic. Longer wavelengths are hotter than,
yes.
Yeah.
And do you have a rationale for why this happens? Like why are...
I'm just a naive, I mean, we're talking about very persistence. No, I don't. I mean, I just painted a picture of it in my mind that makes sense, but I don't have a reasonable physical explanation of this.
So could it be that the longer wavelengths are traveling bigger amplitudes or faster or something?
More bacteria? More bacteria injecting energy in it?
Yeah, but all of this is just pictures that I made in my mind, but I cannot rationalize this like thoroughly.
And it's also very continuous, so it's not like there is some intrinsic length scale associated with the bacteria that's at play either. So seems fascinating anyway.
Yeah, thank you.
The other side.
You might expect that if you had, say, a dislocation or some other structure, in the vicinity of that, the bacteria might actually be channeled in funny ways. Have you ever looked at that? I mean, supposing you have an edge dislocation, a dislocation line here, it's one side is different from another. So, do you see the bacteria move or accumulate in directional ways when there's structural disorder like dislocation?
Yeah, that is a great question. Actually, we're trying to look at the defect motion in general now, both in the and the role that bacteria play in that is definitely, it's a good point that I haven't thought of, that could be affected by the fact that the particles are moving, except of particles being static. Yeah. And
more generally, it would be interesting to look for somewhat more striking signatures of time reversal braking rather than simply a Q-dependent effective temperature. So I was just wondering if you have any in mind. I can think of some. No, if you have any ideas, they're welcome.
Go ahead.
Yes, I'm also very fond of the system. I enjoyed it very much. And my question is, by chance, a little bit related to what Sriram asked. So the question is the collisions that between the bacteria and the particles. Now they are very different from the collisions that generate thermal agitation, right? Yeah. And so they are probably they're more inelastic. Yeah. And so, for example, I can imagine a pathological situation in which all the bacteria swim tangentially to the sphere, and there would be no transfer of momentum in the direction that you're interested in, right? Yeah. Have you ever seen anything like this? Is this too pathological thinking, or?
No, I haven't seen, because I haven't seen bacteria just swimming around and getting trapped. I've seen bacteria get stuck on a particle and then the particle doing crazy stuff. But yeah, it's interesting also to know how, to understand how tau, this persistence time, changes with also bacterial speed, right? Because the lower the bacteria speed, the higher the persistence time seems to be. So we had to play around a lot to find like a high enough active diffusivity and high, because when bacteria go very fast, they swim away from the particle very quickly. So tau becomes very small because the bacteria is not pushing the particle anymore. So it's more then similar to a thermal system than an active one. So yeah, that is interesting. Maybe playing around with fatter bacteria or something
would
be.
If you change the shape of the particles.
Oh yeah, that is interesting. Then yeah, I mean, then if it's known that ellipsoids diffuse differently in active baths than in passive baths. So yeah, that would be interesting.
And thank you.
Thank you.
So I had a suggestion of these beautiful experiments. You've looked at a conventional Lindemann criterion, which is intuitive. It's a little imprecise even for 3D solids, but there's an orientational version of that. If you look at the curl of the displacement field, then you're looking at the wandering around of the local crystallographic axes. I wonder if that might be an interesting thing. And when that's pi, when that turns out to be 30 degrees, you could say, that's my melting criterion. Yeah. And would it agree with the other one? Or have you tried looking at other criteria?
Yeah, that's a good point. No, we haven't tried 3D Lindemann, but we could. I mean, it's a...
Well, you're just talking about a 2D curl that would reflect the local axis.
Yeah.
Okay, thank you.
Thank you.
And we have one last question, and pardon, you can set up before.
Thanks for the great talk. I worked with microbes, so this was inspiring. So you said that this system size is like 100-ish particles, and then maybe
hundreds,
100, and then made me think about the effect of the boundary. And I assume that you just leave the boundary free without any kind of confinement, like in terms of the colloids, like how did large particles.
Sorry.
Please go ahead. Yes.
the whole sample is bigger than this and it's sealed. Otherwise, the particles would end up separating from each other, right? Because you need a confinement. So it's just like a lot of particles and then it's sealed with Vaseline so they don't diffuse away from each other. But yeah, of course, unavoidably in experiments, there are some defects somewhere, some stuck particles. So yeah, we just tried to look at an area that was defect-free of stocks. Yeah.
Okay.
That could have a role. Thank you.
Thank you.
Awesome. Thank you so much.
Thank you.